So this is our floating lecture about neurotransmitters. So at the back of your guided notes packet, you should see a chart all about neurotransmitters. And before we get into that chart, I kind of want to refresh your memory because it may have been a while since you watched our lecture about how a synapse works. So I kind of want to make sure I hit that again really quick before we get into the chart so that we can kind of remember some terms. So remember that where neurotransmitters are going to be released is at the synapse. So when the electrical impulse comes down the cell membrane of the neuron, eventually it's going to hit these calcium voltage-gated channels. So a voltage-gated channel opens when the voltage of the neuron changes. So remember that depolarization changes the voltage of said neuron. So these are going to open and the calcium ions are going to flow inside of the cell. When that happens, they are going to cause the diffusion of the synaptic vesicles. The synaptic vesicles contain the neurotransmitters. And that flow of calcium ions pushes the synaptic vesicles towards the synapse, where they're going to release whatever neurotransmitter they're containing. Neurons are often specific about what kind of neurotransmitter they can make. Sometimes they can make multiple. It just kind of depends on where they're located and like what kind of communication they're engaging in. Remember that neurotransmitters can either be excitatory or inhibitory. And what that refers to is what's going to happen to the next neuron. If it's excitatory, that neurotransmitter is going to cause these channels on the postsynaptic neuron to open and have those sodium ions move in, so depolarization can begin to happen. So the more those neurotransmitters get released, then the more likely we are to see that threshold met and depolarization to occur. If we're inhibitory, what that means is it blocks the channel from opening and prevents the sodium ions from flowing into the cell. So inhibitory get us further away from depolarization, whereas excitatory get us closer to depolarization. And one of the things that we have to keep in mind is that how often these neurotransmitters are used and released into a synapse actually causes the postsynaptic neuron to change shape. And here's what I mean by that. If you have a nerve impulse running down this neuron and constantly releasing these vesicles and releasing these neurotransmitters, constantly stimulating this neuron, the neuron may get overstressed. And so because of that, it can do a couple of things. It can remove some of these channel proteins. So it can remove some of the receptors. So it becomes less receptive to the neurotransmitter. Or it can become fixated, where it actually will try and like make other connections in order to like bypass this synapse. Or it could even, in some cases, like if we have overstimulated the release of a certain neurotransmitter, we can like lose the ability to make it. So we actually can reshape our brains based on the neurotransmitter usage. And this can be one of the reasons that psychological diseases that are based around neurotransmitters are so difficult to treat, is because we are fighting against the brain's own restructuring habits in order to treat these disorders. So those are just a couple of points I wanted to hit before we got into the chart. Let's talk about neurotransmitters now. So this is a breakdown of the families of neurotransmitters. We're going to start here with acetylcholine. Acetylcholine, if you look at your chart, you will see the neurotransmitter name, where it functions, its action, what that function means, and then any special notes I might have about it. So acetylcholine works in two places. We find it in the skeletal muscles, so it works on skeletal muscles, and we find it in the central nervous system. In skeletal muscles, it is exclusively excitatory. In the central nervous system, it's excitatory or inhibitory based on the receptor and how the receptor functions. The function, when we're talking about the skeletal muscles, is that it stimulates the muscles to move. When we're working in the central nervous system, it's similar to any other neurotransmitter, and it works to relay nerve impulses. And the really interesting thing about acetylcholine is that acetylcholine is possibly the oldest neurotransmitter on our planet. We think it is the oldest because it is shared between so many different organisms. In fact, octopi share acetylcholine, and it also contracts their muscles just like it does in us. Now we're going to move into the amines here in the middle. We're going to start with norepinephrine. Norepinephrine works in the central nervous system and in the peripheral nervous system. It is excitatory or inhibitory based on its receptor. And this is the feel-good neurotransmitter. The special note here is that amphetamines, that whole class of drug, enhances the release of norepinephrine. And norepinephrine, when it has that enhanced release, like I said before, your neurons will try to compensate for that increase wave of norepinephrine by removing receptors. They'll remove receptors, and so it will make it harder to stimulate a neuron in order to so that the neuron doesn't get worn out. And so this is why we see individuals who are addicted to amphetamines have to continually increase the dosage that they use, because their brain has physically changed shape in response to the increased amounts of norepinephrine that are being released. On the flip side, if we see a decrease in the release of norepinephrine, that is actually a contributing factor to depression. Talking about dopamine. Dopamine is also found in the central nervous system and in the peripheral nervous system. It is excitatory or inhibitory based on its receptor, and it's another feel-good neurotransmitter. It also is affected by amphetamines. So amphetamines enhance the release of dopamine as well. Another drug also works on dopamine, but in a different way. Remember how we talked about after the release of neurotransmitters, we have to get those neurotransmitters back into the presynaptic neuron, and we call that reabsorption. So once those neurotransmitters have diffused across, activated the postsynaptic neurons, protein channels, they are then going to diffuse back and be reabsorbed into their original neuron. In the case of dopamine, cocaine actually prevents the reuptake or the reabsorption of dopamine. So dopamine is now blocked from being reabsorbed so it can keep activating those neurons, those postsynaptic neurons. Then we have serotonin. This is going to be found in the central nervous system. It is mainly inhibitory, and this is the management molecule. This is going to regulate sleep, appetite, mood, and it can even have an influence on migraines. And with serotonin, the special note there is that by blocking the reuptake, this is how we treat anxiety. So most prescription anxiety drugs block the reuptake of serotonin. Other drugs that also influence how serotonin works are LSD. So LSD blocks the channels that respond to serotonin. And then ecstasy enhances the activity of serotonin. So it enhances the ability for serotonin to trigger synapses. Moving on to histamine. This is found in the central nervous system, specifically in the hypothalamus. It is excitatory or inhibitory based on the receptor. And this is another management molecule. It regulates wakefulness, appetite control, learning, and memory. And this is how the hypothalamus carries out its actions. Remember, we talked about the hypothalamus is that interplay between the brain and the endocrine system. And this histamine is really that bridge that allows it to bridge those two systems. We're going to move on to the amino acids and talk about GABA. It's found in the central nervous system. It's mainly inhibitory, and it is the primary inhibitor of the brain. And blocking the synthesis and release or action results in convulsions. So we can actually have overstimulation of the brain without GABA being present. And that's what the convulsions are the product of. Alcohol, anti-anxiety medications, all augment the functionality of GABA. So when they talk about alcohol being sometimes being like a depressant, this is what's happening, is it's enhancing GABA's functionality in the brain and inhibiting the neurons from firing. This is why consuming alcohol is so dangerous for driving, is because you're actually preventing your neurons from firing, and you're driving a several ton vehicle, and so you do not have the reaction speed simply because your neurons cannot communicate to each other. So this is why driving under the influence is so dangerous, is because your brain is not functioning correctly. You cannot make proper decisions. You cannot comprehend your surroundings because your neurons are completely inhibited because we have increased the functionality of GABA. Moving on to glutamate. Glutamate is found in the central nervous system. It is generally excitatory, and this is used for learning and memory. And then our special note here is that it is highly active during strokes and stimulates the neurons until they die. So one of the problems with strokes is that the neurons are getting so much blood that they start firing their synapses. They start releasing neurotransmitters over and over and over again. And eventually that will cause them so much stress that they will die. Generally, glutamate release is commonly caused by oxygen deprivation because of a blocked blood vessel that is preventing proper oxygen circulation around the brain. Moving on to glycine. Glycine is found in the central nervous system. It's generally inhibitory, and this is the principal inhibitor found in the spinal cord. Strychnine blocks its receptors, which results in convulsions and respiratory arrest. So strychnine is a poison. You might have heard of it in like spy movies, and it works on the spinal cord and causes those neurons to fire so often that convulsions ensue and the respiratory system can no longer function. Moving on to the peptides, we have endorphins. These are found in the central nervous system. They're generally inhibitory, and their main goal is to inhibit pain. So they prevent pain messages from reaching the brain. Morphine, heroin, methadone, all of those mimic endorphins. So they are chemically very similar in structure, so that they prevent those pain receptors from receiving those pain messages. So it isn't that the pain isn't occurring, it's that you no longer perceive the pain. And unlike the other drugs that we have mentioned where they stimulate the release or they prevent the reuptake and your just natural levels are what are causing the change, these are actually chemicals that look the same to the receptors. They aren't exactly the same, but they look so much similar to endorphins that the receptors can't tell the difference. And so it blocks those messages. Next we have tachykinins and substance P. Those two do the same thing, so that's why they're on the same line. We're in the same box together. And they work in the central nervous system and in the peripheral nervous system. And they are excitatory, and they mediate pain transmission in the peripheral nervous system. And in the central nervous system, they control respiratory and cardiovascular action and mood. Next, we have somatostatins. Somatostatins are found in the central nervous system, and they are generally inhibitory. And this regulates the gut-brain information pathway. It also inhibits the release of growth hormone. Then we have cholecystokinin. These are found in the central nervous system. They are generally excitatory, and they're involved in anxiety, pain, and memory. They also are involved in the gut-brain information pathway, and they inhibit appetite. And then we get to the others. We have ATP. ATP is found in the central nervous system and in the peripheral nervous system. It is excitatory or inhibitory based on its receptor, and this provokes pain sensation. Adenosine is also found in the central nervous system and in the peripheral nervous system. It is generally inhibitory. Its functionality is that it dilates the arterioles, increasing blood flow to the heart and other tissues. And it may be involved in the sleep-wake cycle and terminating seizures. Caffeine works on adenosine receptors. It blocks those receptors so that the adenosine can't stimulate, can't block those neurons from firing. So remember, because it's inhibitory, if we block those receptors, it allows more excitatory neurotransmitters to have access to the neuron. So caffeine doesn't necessarily make you feel more awake. It just prevents you from feeling sleepy, essentially. Then we have nitric oxide. This is found in the central nervous system and in the peripheral nervous system. It is excitatory or inhibitory based on its receptor. And this is just a normal communicator. It's just going to be a neural transmitter. If we see an increase in the release of nitric oxide, generally what that relates to is stroke damage. So one of the things that we can check if we are worried that someone has had a stroke is we check to see if nitric oxide levels are increased. If they are, that would probably indicate that we are seeing a patient who has had stroke. Enhancing the action of nitric oxide is used to treat some types of male impotence. So sometimes we need to increase blood flow kind of thing, and nitric oxide can help with that. Then we have carbon monoxide. This is found in the central nervous system, specifically in the brain. It is excitatory or inhibitory, and it communicates between brain neurons. It's just another neurotransmitter. And then the last one we have is the endocannabinoids. These are found in the central nervous system. They are inhibitory, and they are involved in memory, appetite control, nausea, and neural development. And the special note here is that receptors are activated by THC. THC is the active ingredient in cannabis. And you might notice that the things that are involved in are also things that are affected by the use of cannabis. Memory, appetite, nausea, neural development are all impacted by the use of cannabis. This is why one of the ways that medical marijuana is utilized is with cancer patients. What can happen in cancer patients is that you will remember that we talked about mitosis rates being different in different tissues. The lining of your digestive system is highly mitotic. It goes through mitosis very, very quickly. And cancer treatments kill off cells that go through mitosis very quickly. And what that means is you can often have very painful digestive tract issues while you're undergoing cancer treatment. and it just kills someone's appetite. And so people undergoing cancer treatment often lose a bunch of weight, which can be very serious when, the whole point is trying to keep this person alive, and then they're losing a bunch of weight, which can have like systematic problems. And so utilizing cannabis can really be useful to increase someone's appetite so that they will get calories in to keep their body going despite the painful nature of their digestive system at the time. So that's one of those huge benefits. But on the downside, interfering with neural development is extremely troubling. So one of the major concerns we have around cannabis, repeated, consistent cannabis use, is that you actually disrupt the neural development of an individual and affect their memory. And particularly with how potent, how concentrated THC levels have become in modern cannabis, it becomes very concerning, particularly in underage users of cannabis. The concern becomes that basically you have permanently altered your brain by using this. We do see, depending on the age of starting to use cannabis, we can see brain recovery. So after the discontinued use, so like memory problems can kind of dissipate after those endocannabinoid, after the THC has worked its way out of the system, which can take up to 6 months after stopping using cannabis. And because remember, the brain becomes restructured, and so we're having to restructure the brain for now having less like neurotransmitters being used. So just some things to keep in mind about different things. The last thing I wanted to touch on in this lecture was I really wanted to kind of show you some disorders that are associated with each of these classes of neurotransmitters, and how if we have dysfunctions in any of these specific like families of neurotransmitters, what those look like. So just some like things to like pick up on, things like where depression falls, where anxiety falls, those kinds of things, just so you can kind of get a sense of like how important neurotransmitters are for the normal functioning of our nervous system. And that will round out our lecture on neurotransmitters. Possibly this is the end of the nervous system for you, if this was the last one you decided to watch. Remember to study for your test and keep up on things.